Greetings everyone! In this video, we are going to explore the introduction of pathogen, toxic and pathogenic metabolites produced, factors affecting toxin production, mechanism of infection and foodborne diseases. Let's get started.
Our presentation today will start with introduction part that focus on 4 main points. The first one is overview of the pathogen, characteristic, food safety and application. Let's begin with the overview of the pathogen. Overview of the pathogen. Clostridium botulinum or C. botulinum is a gram-positive anaerobic bacterium known for producing one of the most potent neurotoxins. This bacterium thrives in low oxygen environment and form heat-resistant spores, making it significant concern for food safety. A common source of foodborne botulism comes from improperly canned or preserved food, especially low-acid one. This condition creates an ideal environment for the bacteria to grow and produce its deadly toxin. While the toxin is incredibly dangerous, it is also carefully regulated and has medical and cosmetic applications, such as treating muscle spasm, excessive sweating, and reducing wrinkles. However, its administration is strictly limited to qualified healthcare professionals. Let's move on to the characteristic of Clostridium botulinum. The first one is C. botulinum is a gram-positive bacterium with a rod-shaped structure, making it distinct under the microscope. The second one is anaerobic nature. This bacterium thrives in low oxygen environment, which is why improperly sealed or stored food often provide ideal condition for its growth. Next is motility. C. botulinum is motile, meaning it has the ability to move on its own, which enhances its ability to spread in favorable conditions. Next is spore formation. It forms heat-resistant spores that can survive in extreme conditions, allowing the bacterium to persist in environment unsuitable for most microorganisms. It also environmental presence. The spores of C. botulinum are commonly found in soil, rivers, and seawater, highlighting its natural occurrence in diverse environments. Finally, C. botulinum produces botulinum toxin, which is one of the most potent neurotoxins known. This toxin can cause botulism, a serious illness affecting the nervous system.
Moving on, I'm going to talk about the relevance to food safety. Firstly, Clostridium botulinum produces botulinum toxin under anaerobic conditions. It is often present in improperly processed or stored foods. The toxin disrupts nerve function, leading to paralysis and death even in small amounts. Next, it is also connected to foodborne risk. Foods like contaminated food that has low acid is prone to foodborne risk. For example, improper processed canned foods where sterilization was insufficient, vacuum packed or preserved, including smoked or salted fish foods, and improperly processed honey will be linked to infant botulism. Furthermore, food processing and preservation also contributes to the food safety, as the spores are highly resistant to heat, meaning that it can survive standard cooking or pasteurization if not done correctly. Thus, it is important to ensure sterilization during canning processes, like using high pressure or high temperature methods, besides acidifying foods or adding preservatives to inhibit growth, and lastly, refrigeration at below 4 degrees Celsius to slow growth in certain food products. Furthermore, botulinum toxin has several beneficial applications when produced and handled under controlled conditions. In medical uses, the toxin marketed as Botox is widely used in small purified doses to treat various medical conditions like neurological disorders such as dystonia and chronic migraines, hyperhidrosis, excessive sweating, and muscle stiffness. In cosmetic industry, botulinum toxin is used to reduce wrinkles and fine lines by temporarily paralyzing facial muscles. In research, studies on Clostridium botulinum enhance our understanding of neurotoxins and their mechanisms, contributing to fields like neurology and toxicology.
One of the toxin and pathogenic metabolites produced by the Clostridium botulinum is the botulinum neurotoxin. It is a zinc metalloprotease consists of a heavy chain and a light chain linked together by a single disulfide bond. It specifically clips the snare protein in the postsynaptic nerve terminals, which will prevent the release of neurotransmitters and blocking neurotransmission to effector muscles, leading to muscle paralysis. The earliest symptom caused by the botulinum neurotoxin is blur or double vision, dry mouth, and difficulty in speaking. Over time, it can develop into symmetrical descending flaccid paralysis. If the patient leaves untreated, it will lead to respiratory failure and death.
Next, toxin and pathogenic metabolites that produce is spores. Clostridium botulinum spores are a dormant and highly resistant form of bacteria. These spores are incredibly tough, capable of surviving extreme conditions such as high heat and dryness. While they are harmless in their dormant state, they can become very dangerous under the right conditions. In environments with low oxygen, low acidity, pH above 4.6, and warm, these spores germinate into active cells. Once active, they produce botulinum neurotoxin, one of the most potent poisons known, which can contaminate food and lead to a serious illness called botulism. These spores are commonly found in soil, water, and dust, which means food can easily become contaminated during processing or handling. Home-canned foods are especially risky if not prepared correctly. For infants, ingestion of spores can cause a condition called infant botulism, as their immature digestive systems allow spores to germinate in their intestines. Thankfully, botulism is preventable. To stop spores from germinating and producing toxins, we need to maintain acidic conditions in food with a pH below 4.6, keep food cold at temperatures below 10 degrees Celsius, and use high heat, such as pressure canning, to destroy spores during food preparation. By controlling these conditions, we can ensure food safety and prevent Clostridium botulinum spores from becoming a serious threat.
Next, we are going to move to the intrinsic factors that influence the production of toxins by Clostridium botulinum. The first factor is pH level. From my research, toxin production occurs at pH above 4.6. This means acidic foods such as canned tomatoes which have pH below 4.6 are safer from botulism. Next is nutrient availability. Clostridium botulinum thrive in food rich in protein and carbohydrate such as meat, fish, and dairy products. This nutrient-rich food provide the ideal condition for the bacteria to grow and produce toxins. The third factor is water activity, which refers to the availability of free water in food. Clostridium botulinum requires sufficient water activity to grow and produce toxins. Food with low water activity such as salted fish or dry fruits are much safer because the lack of moisture prevents bacterial growth. By controlling these intrinsic factors, we can minimize the risk of Clostridium botulinum in food.
Factors are affecting toxin produce. The first is temperature, stay between 10 to 50 Celsius with optimal growth at 35 to 40 for polyoltic strains and 25 to 30 Celsius for non-polyoltic strains. Refrigeration below 30 can slow toxic production. The second is humidity. High humidity promotes the survival of botulinum spore in the food processing and storage areas. Lower humidity combined with reduced water activity in the food limits toxic production. The third is oxygen availability. C. botulinum is an obligate anaerobe requiring oxygen-free environment to grow and produce toxins.
And now I will explain about the mechanism of infection with the first step, which is insertion. The toxin often enter the body through three main ways, which are from ingestion, inhalation, or infection. Ingestion may occur when food, often canned, is exposed and contaminated by these toxins. Meanwhile, inhalation botulism is rare and usually occurs when the toxin is aerosolized and inhaled into our respiratory tract. And lastly, infection happens when spore contaminates a wound, germinate, and then producing toxins in the wound site. Step 2 is absorption. The toxin is then absorbed into our bloodstream where it then circulates throughout the targeted nervous system. For ingestion, the toxin is absorbed primarily into our small intestine, which then enters our bloodstream via intestinal capillaries. For infection, the toxin thrives in anaerobic condition where it will then generally remain bound inside the wound site. And lastly, for inhalation, after the toxins enter the lungs into our respiratory mucus, it enters the bloodstream and follows the same pathway as the other forms of botulism. Step 3 is binding. Once the toxin has entered the bloodstream, it specifically targets the neuromuscular junction, where it will then bind to receptors on the presynaptic nerve terminal and starts initiating its toxic effect. These toxins specifically target motor neurons that will release acetylcholine, as it is a site with a high activity of acetylcholine distribution. Once the toxins bind to a neuron, it starts into the next step, which is internalization. eventually blocking the formation and release of acetylcholine.
Once the botulinum toxin binds to the nerve terminal, it is taken into the cell through endocytosis, forming a vesicle called an endosome inside the acidic environment of the endosome. The toxin undergoes a structural change, allowing its light chain to escape into the cytoplasm. The light chain then cleaves the SNAP proteins, which are essential for the release of the neurotransmitter acetylcholine. Without acetylcholine, nerve signals cannot reach the muscles, leading to paralysis. This paralysis begins with weakness in muscles, controlling the face, speech, and swallowing, and can progress to affect breathing muscles, causing life-threatening respiratory failure if untreated.
Next, I'm going to talk about the foodborne disease caused by Clostridium botulinum. Clostridium botulinum causes botulism, which is a severe and potentially fatal foodborne illness resulting from the ingestion of its neurotoxins. These toxins are among the most potent known to humans and primarily affect the nervous system. Botulism symptoms include blurred vision, drooping eyelids, difficulty swallowing, and muscle weakness, and also in severe cases, respiratory paralysis. The disease is especially dangerous for populations at risk, including infants, older adults, and individuals with weakened immune systems.
Next, I am going to talk about the causes and risk factors associated with Clostridium botulinum. Firstly, the sources of infection include improperly canned or preserved foods, fermented foods, and vacuum-packed products. These types of foods create an environment where the bacteria can thrive. Moving on to the contributing factors, the growth and toxin production of Clostridium botulinum are encouraged by anaerobic conditions, which are environments without oxygen. Additionally, poor hygiene and improper safety measures during food preparation further increase the risk of contamination. Lastly, let's discuss prevention. To minimize the risk, it is important to ensure proper food preservation and storage methods. This includes maintaining correct temperatures and conditions during processing. Also, following strict hygiene and safety protocols is critical to prevent contamination during food preparation.
In conclusion, Clostridium botulinum is a gram-positive anaerobic spore-forming bacterium that causes severe foodborne diseases through the production of botulinum neurotoxins, which is among the most potent biological toxin. The mechanism of infection involves ingestion of performed toxins in improperly processed or stored foods, leading to neuroparalysis by blocking acetylcholine release at neuromuscular junctions. Toxin production is influenced by factors such as pH level, nutrient availability, water activity, temperature, humidity, and also oxygen availability. The primary pathogenic metabolite, botulinum toxin, is responsible for symptoms ranging from muscle weakness to respiratory failure. Effective control of this pathogen relies on proper food preservation, stringent hygiene, and awareness of conditions that promote its growth and toxin production. That's the end of our presentation. Thank you.